Hey guys, Death Angel here, and I just put up that video from the nurse, or I'm sorry, the doctor, that was trying to explain the nanoparticles and the dangers of the new vaccine. Well, here's the actual science behind it, and we're going to get started. And you'll recognize how everybody has told you about how exosomes are the garbage of the cells. Well, it's much, much more than that. RFB, I know for sure, has told everybody just the garbage of the cells. Well, the research is right here. This is from the Journal of Nanobiotechnology, so it's going to be a little wordy, but I went through ahead of it before I recorded this. This is my third time recording it. I lost the original video when I got banned last time, but here we go, and this is going to be the real science. Background: exosomes are considered key elements for communication between cells, but very little is known about the mechanisms and selectivity of the transference processes involving exosomes released from different cells. Result: in this study, we have investigated the transfer of hollow gold nanoparticles, HGNs, between different cells. When these HGNs were loaded within exosomes secreted by human placental mesenchymal stem cells, MSCs, these HGNs were successfully incorporated by the MSC's exosome biogenesis pathway and released as HGNs loaded exosomes, meaning your body took it up and released it again with a new cell coding, so your body would recognize it while it had nanoparticles inside of it. This document will also show photos of the actual exosomes under the electron microscope, so you can see them, what they really look like on a nanoscale. In this study, we demonstrate the selectivity of in vitro exosomal transfer between certain cell types and how this phenomenon can be exploited to develop new specific vectors for advanced therapies. But you'll see below they can also deliver viruses, anything they want. The body of an adult person contains around 37 billion cells that function coordinately. To work as a whole entity, many coordination mechanisms coexist, using different factors as messengers. For example, the nervous system makes strong use of communication by electrical impulses, and the endocrine system is capable to send messages to distant areas mediated by hormones. One of the most intensely studied at the moment concerns the exchange of genetic material and proteins mediated by the exosomes or microvesicles secreted by the cells. Many cell types present in the organism release vesicles of different nature, including apoptic bodies, ectosomes, microvesicles, and exosomes. Exosomes were known since 1981, when Trams and co-workers defined exosomes as vesicles derived from an exfoliation of the plasmatic membrane, although the term exosome was coined in 1987. Early studies considered exosomes as garbage of the cells. This is what you're being fed by everybody, because that's what they were told to tell you. Even though it was known that they contain genetic material, including mRNA, miRNA, DNA, and proteins, so does the vaccine. Eventually, it was discovered that exosomes not only could serve as a mechanism to discharge unwanted material from cells, but also could form the basis of an efficient cell-to-cell communication mechanism. Valetti showed that exosomal mRNA and microRNA could be transferred to another cell, being functional. Recent works dealing with the properties and functions of cell-derived exosomes suggests that they are involved in a variety of scenarios, including central nervous system diseases, myocardial ischemia, circulation damage, liver and kidney injury, and the modulation of tumor hallmarks, inducing angiogenesis and metastasis. So they're even responsible for metastasizing cancer. This is how amazing your body is. It's been kept from everybody. Excuse me. Studying exosomal transfer between cells can provide key information on the evolution of different diseases. They also hold promise as a tool for allowing early diagnosis, since exosomes are present in most biological fluids, blood, urine, saliva, sperm, etc., and therefore a variety of tests can be developed. Another highly important characteristic of exosomes relates to their role as transference vectors of membrane receptors, functional proteins as growth factors, or nucleic acids. Right here. If the specific exosome-based transport could be controlled, it could be potentially used as to transfer therapeutic elements, drugs, viruses, nanoparticles, etc. But they've already done it, and it's already here in the document. In fact, some investigations have already explored this path, harvesting exosomes and loading them with the desired therapeutics. But as you'll see below, just like that lady was, I don't remember I think it was Luciferase because it used light, they've already done it. This is the Luciferase. It's all right here. However, electroporation and sonication can disrupt the exosomal membrane, and therefore other routes that exploit natural uptake mechanisms are preferred. And right there, exploit natural uptake. Your own body against you. Right here. However, in spite of the fact that most proposed applications focus on the ability of exosomes to target specific cells or tissues, the understanding of their selective trafficking and communication mechanisms remains poor. Some of the few works on this matter suggest that extracellular vesicles, including exosomes, can be incorporated by every cell type studied, which is a big deal because when you normally get, like, say you got a urinary tract infection, that's why it only affects that area and not your whole body. These can be used on any cell in the body. Herein, we have used relatively large entities, 40 nanometers in diameter, hollow gold nanoparticles as tools to monitor the exosomal transference between cells. HGNs behave as efficient transductors of light into heat, that's the luciferase, and can be used to destroy malignant cells by hypothermia or to trigger other temperature-driven processes, including drug delivery and gene expression. Gene expression being the S gene mutation that I put in the coronavirus patent. It's in the truth video with all the documents listed. We have successfully incorporated HGNs into the MSC exosome biogenesis pathway. Using HGN-loaded exosomes, we were able to follow cell-to-cell communication, and in particular, how the exosome's cellular origin could affect HGN's transference between cells. Right here, hollow gold nanoparticles. The HGNs were hollow pseudospheres with a thick shell. A uniform shell of almost 5 nanometers could be observed coating the HGNs. And it is very wordy, so I'm trying to skip over what I didn't highlight. I'll leave the document like I always do for you to read. And it has the images in the document as well. It shows you the shells they're talking about right here that they coat them with. 5 nanometers thick. Yeah, right here, characterization of HGNs. That's hollow gold nanoparticles right here. And how they trick your body into taking it up. Furthermore, we also confirmed that the presence of both types of nanoparticles in the cell cultures did not produce significant changes on cell cycle phases at the sub-cytotoxic doses, meaning that it didn't change the cell, didn't kill the cell. NPs inside MSCs was evaluated by confocal microscopy and was indirectly quantified by MPAES, using the total amount of gold measured inside the cells. The results indicate that significantly more PEG-HENs were localized inside cells compared to the amounts retrieved for HENs, probably due to the higher stability and the reduced agglomeration in the culture media provided by the PEG surface functionalization. HGNs followed in the process of being incorporated into target cells to verify if those strategies were compatible with the exosomal pathway. This is the pathway they're using to exploit to deliver their BS. A spherical shape and characteristic lipidic bilayer membrane was observed in most of them. Dynamic light scattering, DLS, and nanoparticle tracking analysis measurements gave a mean particle diameter around 180 nanometers in their PBS test. Just like the doctor was saying, that the light in the body that you can't even see, the merging of blue and red fluorescence, which corresponds with aggregates of nanoparticles and exosomes or late endosomes co-localization, indicating the multivascular bodies within the cell already contain HGN-loaded exosomes. And here they are under the microscope. See 100 nanometers, 50 nanometers, I think one of them goes down to 20 nanometers, but that's what they actually look like. Little tiny biowarfare delivery systems against everybody. And here's another chart of them glowing under the cell culture. There's your luciferase that the doctor was talking about. And I'm not, maybe I was wrong to make fun of her a little bit, but we already knew this information. This is the third time I've made this video. Right here, exosomes. Nanoparticles right here. And this is where they merge. And this is the zoomed in. It's all included. The real science, the actual information. And here they are again. Yeah, there's the 20 nanometer one. Location of PEG, hollow gold nanoparticles. That's what they look like. I missed a spot. I gotta go back up real quick. Sorry, guys. After these were excreted, the HGN-loaded exosomes could be easily recovered by ultracentrifugation and subjected to TEM analysis. TEM images of purified exosome from MSC culture supernatants after incubation with PEG hollow gold nanoparticles showed that most exosomes were loaded with at least one nanoparticle, confirming the high yield of nanoparticle loading into exosomes. These values confirmed that the presence of the nanoparticles inside the exosomes did not affect their endogenous diameter, meaning it didn't change the size and the shape of the cell. And right here, here's more nanoparticles trafficking between cells. You can see right here the actual data, real science. And you can see where they use dyes to mark the different cells, the luciferase. Exosome has specific vectors for PEG-HGNs between different cell lines. Given the way nanoparticle-loaded exosomes are generated, we postulated that exosomes derived from a cell line are fingerprinted with hallmarks of that cell type, and therefore could be preferentially uptaken by the same cell line, even under co-culture conditions with other cell lines. MSCs and monocytes, they were significantly uptaken by both cell types between 1 and 2, gold/cell population. We observed that nanoparticles transference occurs preferentially between MSCs, and only a minor fraction of PEG-HGNs were captured by monocytes, both at 24 hours and 48 hours. It is reported that when monocytic cells recognize nanoparticles in the media as efficient scavengers, they firstly internalize NPs and trap them in the exosomal pathway, releasing those materials again to the media. I have to start over. I have to edit these two parts together. I had to take a break for a second. It is reported that when monocytic cells recognize nanoparticles in the media as efficient scavengers, they firstly internalize nanoparticles and trap them in the endosmal exosomal pathway, releasing those materials again into the media. Nanoparticles that either were present originally in the medium or were excreted through a non-exosomal pathway and therefore lack the cell-specific exosomal coding. This is very important because they have to get the cell to take it up and then spit it out again so that it coats the nanoparticles in the same proteins that the other cells will recognize, or else your body will recognize it and kick it out. That's how amazing God made us. To validate the conclusions reached with other cell types, the same study was carried out using monocytes and tumoral cells. It showed that separate cultures of each cell, the uptake of PEG-HGNs by monocytes were significantly 5 times higher than that obtained for the tumoral cells. When cultured together, a similar uptake ratio is observed for both types of cells. So even if you have a tumor already in you, same thing. A high amount of nanoparticles were present inside the MSCs, and those were very active in transferring nanoparticles between cells. As you can see in the photos that are included in the document, they already have it. They've already tested it over and over again. Therefore, a specific signature of the exosomes released from a cell type is conserved and used as a recognition moiety for the same type of cell, which is exactly what I just said. It coats the nanoparticle in its specific protein that the other cells of the same type will recognize, so that the other cells will take it up immediately, without your body recognizing it as being an invader and kicking it out. As a proof of concept of the possibility of inducing selective cell death using high intrinsic selectivity of exosome-mediated transport, we analyzed the in vitro photothermal effect, there's your luciferase again, of HGN-loaded exosomes, originated from the MSCs on separate culture containing MSCs melanoma cells, and resulting HGN-containing exosomes were harvested and purified. So unlike COVID, this has already actually been done and harvested and purified. After washing, the cell cultures were irradiated. I'll go back up to this in a second. With an NIR laser, and viability toxicity was evaluated, flow symmetry, as well as direct live dead staining, which is up here. Which cells survived, which died. And they got it down to a science right here. Photothermal effects mediated by MSCs, derived exosomes containing plasmonic hollow gold nanoparticles. And you can see it for yourself, the real data. To this end, MSCs previously loaded with PEG HGNs were co-cultured with B16F1 cells during a 48-hour period, sufficiently long to have multiple excretion/uptakes of nanoparticles by the MSCs. Thanks to the intrinsic size of nanoparticles and considering the size of the cellular components, and that most of the biological processes occur on the nanometer scale, a novel and promising field has been opened. Thus, research on exosomes and nanoparticle combination has gained attention over the last decade, which is what we said the code. And I guarantee you the 5G, the frequency, all ties into this. Several works have reported the unique properties of HGNs have sufficient transductors of light into heat. And the heat is what kills the cells. There's light again. There's your luciferase again. Being used to destroy malignant cells by hypothermia or to trigger other temperature-driven processes, such as drug delivery or gene expression. The S gene that I warned of in the S gene mutation document. Herein, we study the selectivity of vitro exosomal transfer between certain cell types and how this phenomenon can be exploited to develop new specific vectors for the transmission of relatively large entities, 40 nanometers in diameter, of hollow gold nanoparticles between different cells, when these nanoparticles were loaded with exosomes secreted by human MSCs. They're not studying animals, they're studying humans, us guys. Amongst the characteristics of those MPs, the presence of the SH PEG coding, usually employed right here. This is what I was telling you about a couple times already. Amongst the characteristics of those nanoparticles, the presence of the SHPEG coding is usually employed to provide a stealth cover to retard detection and removal of MPS macrophages by reducing the protein corona formation around the HGNs. Also, both nanoparticle dispersions have a maximum resonance peak of 808 nanometers, in which the absorption of incident light is minimized in biological fluids because of the reduced light absorption by water and chromospheres. This is the coding I'm talking about. If it doesn't go inside the cell and then get spit out again, it doesn't get coded by that coding, and the body will recognize it and get rid of it. Regarding the cytotoxicity test and cellular cycle analysis, our results indicate that the presence of both types of nanoparticles did not produce significant effects on cell viability and on cell cycle distribution at the studied dose. They already have the doses already studied. Right here, this is how sometimes it doesn't work with all cell types. Also, even for the same type of nanoparticle, the internalization mechanisms may be different for different cell lines. That's that coding they need. As previously shown, an intense production of exosomes loaded with PEG HGNs was observed by CD9 and CD63, expression by the BCA analysis and by NTA results. Lab tested and approved. Thereby, we have taken advantage of the intrinsic exosomal biogenesis process to obtain exosomes loaded with gold nanoparticles. This loading process avoids harsh treatments such as membrane rupture by electroporation and chemical bonding on the membrane wall, and is likely to preserve better the natural characteristics of the produced exosomes. They can also put iron inside of them. Derived exosomes loaded with 5 nanometers of iron nanoparticles. Iron definitely responds to electricity or frequency. Other authors have employed more gentle or natural methods to load nanoparticles into exosomes. Our results are in agreement with previous work suggesting that the exosomes have the ability to target specific cells and to serve as selective vehicles. Vehicles for anything they wish to put in there. Right here, nanoparticles are particularly attractive in a scenario of therapeutic nanoparticle delivery, since their loading with exosomes would help to avoid in vivo recognition by the MPS. That's your body's natural defense mechanism. Our results again confirm the high specificity of exosome uptake depending on the exosome origin. This is the first work in which a specific transfer of exosomes is employed for selectivity applied optical hypothermia therapies. I just had to check and make sure I didn't highlight anything else over there. And here's the conclusion. The encapsulation of PEG-HGNs in exosomes using the intrinsic biogenesis pathways of MCS or MSCs allowed us to confirm the high specificity of exosome-mediated cell exchanges according to the cell origin. A wide study has been carried out with cells of different lineage, including stem cells, immune system cells, and tumor and metastatic cell lines. As a proof of concept of exosome-mediated selective transfer of therapeutic material into the targeted cells, for the first time our study demonstrates the specific in vitro optical hypothermia therapy based on exosome delivery of NIR-responding plasmonic nanoparticles. And it just scrolls here to the end. I skipped over a lot. Like I said, it's a very wordy document. This is the real data. And you can come down here and find the author details, acknowledgments, publisher's notes. Here's all the references on the lower right hand side. I'm working on trying to find these videos right now. It won't load up from the document itself, but I'm going to try to find them. And right here, the results have expressed as a mean standard deviation were carried out in triplicate. So they've reproduced this over and over again. And on the right hand side of the screen, you will find links right here to all these references, to all these different studies. 1981, going all the way back. And that's why I said, well, the doctor's information is good, and here's even more studies. I mean, there's over 50 so far. We'll keep going. Looks like 45. And while that doctor's video was good information, it's not the real information. This is the real information. And while it's very hard to understand, once you read it a few times, you'll get it. It's not that hard. Once you read it a couple times. The first time you read it, you'll just get like mind mush. You know what I'm saying? Like you just go numb. But once you do it again, come back to it again a couple times, you'll get it. But other than that, that's about it. Death Angel out. And as always, I'll include the document in the description. And here's the real data, guys. See ya.